Hello, and welcome to the GRACE podcast series. My name is Denise Brock, and I am the Operations Director for the Global Resource for Advancing Cancer Education, or GRACE. In this podcast series, we interview patients, advocates, and healthcare professionals to provide the most updated information for our community and to highlight important issues facing those dealing with a cancer diagnosis. We hope you find this information valuable. For questions or comments, please visit us at cancergrace.org.
Hello and welcome everyone. My name is Dr. Medhavi Gupta. Today we'll be talking about treatment options in metastatic triple-negative breast cancer. Before we talk about the specific treatment options, let's talk a little bit about triple-negative breast cancer. So in today's time, the way we define triple-negative breast cancer is based on what it is not rather than what it is. In the world of breast cancer, we test all the tumors for three specific receptors: estrogen receptor, progesterone receptor, and HER2 receptor. So triple-negative breast cancers lack expression of any of these receptors. They account for approximately 15% of all breast cancers. They are more common in younger women, especially women less than 40 years of age, African American women, and patients who have BRCA mutations, especially BRCA1 mutation. These tumors have aggressive biology and therefore have a worse prognosis as compared to other breast cancer subtypes. They tend to relapse early, especially within the first 3 years, and they are more commonly associated with visceral mets. When it comes to the treatment of metastatic triple-negative breast cancer, it is important to keep few things in mind. Firstly, whether the tumor is PD-L1 positive or not. The reason this information is important is because PD-L1 positivity determines whether the patient is a candidate for immunotherapy or not. Secondly, we see whether the patients have either germline BRCA1 or BRCA2 mutations, because if the patients have either of those two mutations, they could get a class of drugs called as PARP inhibitors. Thirdly, if the patients previously had localized triple-negative breast cancer, it is important to see what were the previous lines of therapies that the patients got, because that will determine the treatment in the metastatic setting. Last but not the least, we see what is the functional status of the patient, because that can help us in estimating what therapies the patients can handle or not. Historically, the treatment options for metastatic triple-negative breast cancer were very limited. But in the last few years, we saw addition of several new drugs. To talk about those agents, we have a very special guest today, Dr. Aditya Bardai. Dr. Bardai is a breast medical oncologist at Massachusetts General Hospital. He is also an assistant professor with the Harvard Medical School. Dr. Bardai, thank you for joining us today.
Thank you for having me.
Let's talk about chemotherapy. NCCN has a number of chemotherapy agents that are recommended for triple-negative breast cancer. Many of these agents have been around for decades. But now the field is moving towards a totally new class of drugs known as antibody-drug conjugates. Dr. Bardai has led the seminal trial, ASCEND trial, and we are really lucky to have him here to talk about the data himself. So in April 2020, FDA approved the sacituzumab govitecan for patients with metastatic triple-negative breast cancer. Dr. Bardai, could you please first tell us, how does this drug act?
Yes, sacituzumab govitecan is a novel antibody-drug conjugate that targets TROP2. TROP2 or trophoblast 2 is an antigen that's overexpressed in majority of breast cancers, including triple-negative breast cancer. So this is a first-in-class antibody-drug conjugate against TROP2. The antibodies against TROP2, it has a linker, which is a hydrolyzable linker. And this is linked with SN38, which is the active metabolite of irinotecan. And the overarching idea is to deliver high doses of SN38 or toxic payload to the cancer cells and the tumor microenvironment as compared to what you can do with chemotherapy. Now, because of the hydrolyzable linker, when the ADC reaches the cancer cell or in the tumor microenvironment, it can release SN38. So even if the cells are TROP2 negative, it can impact those cells because of this bystander effect. So if there is TROP2 heterogeneity, the ADC still has activity because of this bystander effect.
Could you please walk us through the trial that led to the approval?
Yeah, the approval of sacituzumab govitecan was based on a phase 1, phase 2 trial, which demonstrated a response rate of 33% in pre-treated patients with metastatic triple-negative breast cancer. That was a single-arm study, and based on the single-arm study, the FDA granted accelerated approval, given the unmet need and high clinical activity. But this was contingent on the confirmatory trial, which is the ASCEND trial. It was a trial very similar to the phase 1, phase 2, looking at patients with metastatic triple-negative breast cancer in the third line and beyond setting. But the difference was this was a larger trial and also randomized. Patients with metastatic triple-negative breast cancer in the third line and beyond setting were randomized to receive sacituzumab govitecan versus chemotherapy of physician's choice, which could be eribulin, vinorelbine, gemcitabine, or capecitabine. In terms of study results, it was a positive trial. We presented the results at ESMO last year. Median progression-free survival with sacituzumab govitecan was 5.6 months, as opposed to 1.7 months with standard chemotherapy, hazard ratio of 0.41. Similarly, improvement in overall survival, 6.7 months with standard therapy versus 12.1 months with sacituzumab govitecan, a hazard ratio of 0.48. And strikingly, the response rate with sacituzumab govitecan was 35%, very similar to what was seen in the phase 1, phase 2 trial, as opposed to 11% with standard chemotherapy. So these results, A, confirmed what was seen in the phase 1, phase 2 trial, B, demonstrated that sacituzumab govitecan is associated with improvement in both progression-free survival and overall survival in metastatic triple-negative breast cancer. And third, also again highlighted how bad chemotherapy does in this setting, progression-free survival of 1.7 months and overall survival of 6.7 months with standard chemotherapy. So we clearly need to do better for our patients with metastatic triple-negative breast cancer.
That's really exciting data, especially as you said that patients who have already received therapies in metastatic triple-negative breast cancer, they tend to do poorly. So it's really reassuring to have a drug that is actually extending the overall survival. Could you also tell us a little bit about the side effect profile of this drug?
And the side effect of this drug essentially is from the toxic payload, which is SN38. The common side effects that are seen with SN38 include myelosuppression, anemia, neutropenia, thrombocytopenia. So all these side effects are seen with sacituzumab govitecan as well. Diarrhea is the other common side effect. The incidence is much lower as compared to what we see with irinotecan, but certainly a side effect that is seen. And then finally, the drug tends to cause alopecia.
So we spoke about all the new therapies, especially the most recent ones. Triple-negative breast cancer, especially in the metastatic setting, has been a really challenging tumor type to treat. Dr. Bardai, why do you think that is the case?
Yeah, I think triple-negative breast cancer is tough to treat, first because of the definition of triple-negative breast cancer. Essentially, it's a diagnosis of exclusion. It's a mixed bag. It includes tumors that are basal-like, but also luminal-like. So I think we need a classification that's much more biology-driven. The second is the significant amount of heterogeneity that's seen, particularly with the basal subtype of triple-negative breast cancer. So if you have a targeted therapy that's super targeted because of heterogeneity, that might be a problem. But if you have a therapy that's very diffuse like chemotherapy, you just cannot get enough toxic payloads in the cancer cells without causing a lot of toxicity. So you need an agent that provides both some degree of heterogeneity so it can address cells that are different, but at the same time not too toxic. And I think the third thing that is important to target triple-negative breast cancer is the need for combination therapy. In general, particularly for ER-positive disease, we tend to use single-agent chemotherapy. But in triple-negative breast cancer, we might need combination therapy. Now, I'm not suggesting combination chemo, you also have a lot of side effects when you try to combine different chemotherapy agents. But I think with antibody-drug conjugates, immunotherapy, as we develop combination therapies, we can improve the therapeutic index, control the disease without causing a lot of side effects.
Well, thank you, Dr. Bardai, for being here with us today and guiding us through the different treatment options.
Thank you.
Thank you again for joining us. This podcast was made possible by the generosity of sponsorship from our friends at Lilly and Exelixis. Please like and follow us on Facebook and Twitter. Send us feedback, share your story, donate, and visit us for more information at cancergrace.org. Thank you for listening.